Alright, so we're going to start up now with our chapter on plant reproduction, and also as we talk about plant reproduction, we're looking at both sexual and asexual plant reproduction. So the first thing we're going to look at, and I should mention too that we're focusing only on flowering plants. We're not looking at gymnosperms, we're not looking at ferns or anything like that, mosses, those kinds of things. We are just looking at the flowering plants, right? So when we look at flower structure, we have different parts of a flower, like we see in this picture, all of which were originally actually leaves. These are modified leaves that have taken on the role of sexual reproduction and perform either the role of pollen production or egg production and the dispersal of those structures as well. Some of the terms we can think about here: a complete flower is a flower that has all four flower parts. Complete flowers have petals. So in this picture here we have petals. They have sepals. They have what's called a carpel. The carpel consists of this entire structure here. So the stigma, the style, the ovary, and the ovules inside of that are part of the carpel. And then also have the stamens. And stamens consist of two parts, the anther and the filament. So if a flower has all four flower parts, petals, sepals, stamens, and carpels, we say it is a complete flower. If it's missing one or more of those different flower parts, we say it's an incomplete flower. And different species vary. Different species will produce complete versus incomplete. Some species might also produce perfect versus imperfect. Perfect flowers are those that have both male and female parts. So in other words, they would have both the stamen and the carpel. Whereas imperfect flowers are either male or female. So in other words, they would have either a stamen or a carpel, but they would not have both. So when we look at the collective term for all the sepals, we would call it the calyx. So in this case, we have in this flower a sepal here, sepal over here, very likely one kind of behind all of this, and then one that would have been present had this part of the flower not been removed. Collectively, all those sepals together are called the calyx. If you look at all the petals together, that is a structure you would call the corolla. So the calyx is for the sepals, and the corolla is for the petals. The andrecium is another name for all the male parts of the flower. Andrecium consists then of the of the anther and the filament. The andrecium consists of the anther and filament. So what we call the stamen, anther, filament, anther's at the top. Filament's like a stalk that connects the anther down here to the bottom. This entire structure here would be called the andrecium, or just the stamen, which is a much easier kind of word to pronounce and obviously to spell as well. So it consists of both the filament and the anther. The gynecium consists then of the female parts of the flower, for those that have female flower parts. That's going to consist of the carpel, which again is the stigma, the style, the ovary, and the ovule inside the ovary. So over here we have a carpel. Another name for that would be the gynecium, and again this consists of a stigma and a style, as well as the ovary and this structure inside of here, which would have been the ovule at one point in time. So those are there as well. Over evolutionary time, there are some trends that we see that happen, typically as plants evolved, as they became more advanced and more complex and became more diverse in terms of a group. There were some general trends in flower evolution that we see. Number one is that flower parts often become fused together. So what initially might have been many, many separate flower parts in a very primitive or ancestral sort of plant form are fused together into singular structures in more modern day and more complex plants. We also would take a look at the loss or reduction of flower parts. Typically, very, very old primitive plants have tons and tons and tons of flower parts. More modern day plants tend to have a loss or reduction of sepals or of petals or of stamens or of carpels, for example. They're missing oftentimes one or more of those flower parts in more modern day plants. A lot of other modifications often interact with pollinators. So whereas the more primitive, the very, very old prehistoric kinds of plants were very often wind pollinated, and there are some like that still even today, as time went on, as animals evolved kind of along with the plants, there became this really tight, close-knit relationship of the plant-pollinator sort of association. So a lot of modifications are there to help pollinators do their job better to ensure the plants are able to get fertilized. Now, this is the first time we've seen the term called alteration of generations, and this is something that plants do that's very, very different from what any animal does. Their life cycle is exceedingly different from the animal life cycle, and this is oftentimes hard for people to kind of wrap their minds around because it's so different from what we're used to thinking about, right? So with alteration of generations, what you have is an organism that goes through both a haploid and a diploid part of their life cycle. They go through a part of their life cycle where you've got cells with single sets of chromosomes, and they spend part of their time in a life cycle where they have two sets of chromosomes. And animals are the same way. If you look at an animal life cycle, everything from beyond the zygote up until gametes are produced. So the embryo, the fetus, the infant, the child, the parent, the adult, those are all multicellular, and all those stages are also diploid. And the zygote actually is also diploid, although it's not multicellular. Once you go into the gamete stage, the gametes, and we're talking there sperm cells and egg cells, are haploid. But in an animal, those gametes are only single-celled, right? So sperm cells, egg cells, those are single cells. They are haploid, but they are single cells. Now, if we kind of shift for a second and talk about plants, plants have both a multicellular haploid form and a multicellular diploid form. Both forms, haploid and diploid, single set versus double set of chromosomes, both forms are multicellular, which is very, very different from animals. Because for us, one of our forms, the haploid form, is single-celled. So it's very, very different. We have, in the case of a plant life cycle, a diploid stage called a sporophyte, and a haploid stage called a gametophyte. And then at that point, you would then return back to the sporophyte stage. When you look at different groups of plants, seedless non-vascular plants versus seedless vascular plants versus gymnosperms versus angiosperms, the emphasis on the different parts of the life cycle is one of the key differences among the different groups. So for example, in the mosses, right, in their group, the gametophyte is the dominant form. It's the biggest, most obvious, around for the longest, and most independent part of the life cycle. Sporophytes completely dependent on the gametophyte for everything. If you look at the ferns and some of their relatives, they actually have independent sporophytes and gametophytes. They're separate from each other in different places, different parts of the environment. But they're, again, both multicellular. And if you look at gymnosperms or angiosperms, their sporophyte is the dominant stage, and their gametophyte is actually microscopic. So the relative emphasis on any given part, haploid, diploid, gametophyte, sporophyte, depends on the group of plants you're looking at. In angiosperms, which is of course what we're focusing on most here in this part of the chapter, the gametophyte is microscopic. It is initially part of what we'll see as the pollen grain as well as the ovules, very, very small structures. Right? So we have initially in the male gametophyte, we have pollen grains, and we'll take a look at how those are produced and what they look like, how they function as well. The female gametophyte is the embryo sac, which initially starts off as an ovule. The embryo sac is much more complicated than the pollen grains are, so we'll take a look at the overall structure of that as well. Okay, so here's our gamete production. We have pollen grains being produced in the anther. So again, this is our same flower from before. We've got anthers up here, connected by filaments down here to the base. If you were to cross-section this anther and look at it head-on, you would see this structure. These are called pollen sacs inside of the anther. So all these little pollen sacs have inside of them what are called microspore mother cells that go through meiosis to produce microspores. And these are now haploid. Right? These are haploid microspores. These microspores can then eventually go through mitosis to kind of amplify their numbers or multiply their numbers so the plants produce tons and tons and tons of pollen. Meanwhile, if we take a look down here in the ovary of the plant, of the flower, the female part of the flower, we take a look at what initially starts off as a megaspore mother cell. It goes through a round of meiosis to produce 4 haploid megaspores. 3 of these guys actually just go away. They disappear kind of at random. More or less, the first 3 are the ones that go away first. This one up here at the top is left behind. So this one survives, and then it goes through mitosis as well, just like our cell up here went through mitosis. This cell goes through mitosis, and it's going to produce a grand total of 7 cells that contain 8 nuclei. And that structure that contains the 7 cells with 8 nuclei is also called the embryo sac and also serves as the female gametophyte. Okay, so if we take a look down here, we have 3 antipodal cells, which don't really do much at all in terms of fertilization. We have 2 cells down here. These 2 little orange ones are called synergids, and their job is to kind of block off this entranceway for pollen grains and for sperm trying to get in. We have the egg cell itself, which is the blue cell. So there's 6 cells. 1, 2, 3, 4, 5, 6 cells, and then one great big cell in the middle. That's cell number 7. And this great big cell in the middle actually has 2 nuclei. So that's why we say the embryo sac consists of 7 cells comprising 8 nuclei. And so eventually, once we have our sperm cells fertilizing the egg cells, we get back to the point where we have a zygote and an embryo, and that kind of restarts us back to the beginning of the life cycle. But again, what we want to think about here is how animal life cycles differ from plant life cycles in terms of haploid and diploid, but especially in terms of single-celled versus multicellular. Okay, so with the anthers now forming pollen grains, microsporangia are involved in each anther, involved in producing what are called microspore mother cells. And again, these are diploid. These are not pollen grains. These are not haploid yet. They're not to the point where they can participate in fertilization. Those microspore mother cells go through meiosis to produce 4 haploid microspores. Each one of these diploid mother cells goes through meiosis to produce 4 haploid microspores through meiosis. From there, the microspores go through mitosis to develop into a pollen grain. These microspores divide once to form two cells, which then become the functional parts of the pollen grain itself. Those two cells consist of what's called a generative cell. The generative cell is the one that's going to eventually go through another round of cell division to make two sperm cells. And it's also going to produce a tube cell, which eventually develops into a pollen tube. So we have the progression here. This is like a flowchart sort of situation. You've got microsporangia produce microspores. Microspore mother cells go through meiosis to make microspores, which are haploid. Those then divide through mitosis into pollen and produce a generative cell and a tube cell, which can be evolved in fertilization. Now we take a look at the embryo sac, female gametophyte. We have the diploid megaspore mother cell producing 4 haploid megaspores. So just like we saw before, we have these megaspore mother cells going through meiosis to make megaspores. Over here we had microspore mother cells going through meiosis to make microspores. Now it's the same thing, but it's involving megaspores. And this again is the point at which only one of those 4 cells can survive. The other 3 are going to degenerate. They're going to go away, leaving behind only the one megaspore left behind. That megaspore then gets bigger, it grows, it goes through mitosis, and it produces, again, those 7 cells with those 8 nuclei like we talked about before. The 3 antipodal cells, 2 synergids, 1 egg, and that one large central cell with 2 nuclei. And so that now is the embryo sac, 7 cells that consist of 8 nuclei total. Now, pollination is kind of the next step. Some plants are what we describe as being self-pollinated, where if pollen from male part of a flower lands on a female part of a flower, fertilization can occur, seeds can be produced, embryos can be produced, and the life cycle continues. Cross-pollination is where pollen from a male flower or from a male plant travels to a female flower or a female plant and causes fertilization. Like I talked about before, a lot of the really early seed plants were wind pollinated because there simply weren't a lot of animals around on land at that point in time to take advantage of as pollinators. But as time goes on, evolutionarily what occurs is that more and more plants become pollinated by animals. It's a much, much more efficient way to have animals moving your pollen from plant to plant than releasing just a ton of pollen into the wind and hoping that at least some of it gets to where it needs to go. We also see, like I talked about before, floral morphology. Forms in flowers co-evolve with pollinators. We see all kinds of markings and shapes and structures and even deceptive coloration to try to get pollinators to do their job with the least possible return for the plants themselves, right? So basically it's a way for plants to take advantage of animals, use them to their benefit for pollination, and in return the plants may not provide anything really at all to the animals. In the best case scenario, they might provide a little bit of nectar to the animals, which might convince them to come back again when it's time to pollinate. We take a look at plants that are insect pollinated, especially those that are pollinated by bees. Bees actually have a really nice ability to see into the UV part of the spectrum, which is something we can't do as humans, vertebrates. We cannot see UV light, but bees can. So a lot of times you'll take a look at a flower like this one that, you know, it's a nice looking flower, but if you expose it to black light, for example, if you bombard it with UV light, then what you see is something completely different. There's this dark circle in the middle, which is like a bullseye for a honeybee. It shows the honeybee where it needs to go in no uncertain terms to find the pollen and to eventually find perhaps nectar as well. Pollination by birds and butterflies is often a little bit tougher because birds and butterflies are a bit heavier, especially the birds. But even in this picture, we see something called a hummingbird moth, which is the size roughly of a hummingbird. And so for flowers pollinated by large animals like these, they're often going to have a landing platform, a place to support the animal so it can rest while it's feeding, rather than spending a very short time and then flying away. A lot of times, flowers pollinated by butterflies are white. Or actually, more in the case of moths, they're white because white shows up best at night, right? It doesn't do you any good to produce bright red or bright blue flowers at night because you can't see them anyway. If it's a moth pollinated or pollinated by a nocturnal animal, you're much better off to use white as a way to attract your pollinators because white can be seen better at night than pretty much every other color. There's no point in producing a lot of pigments because they're not going to do you any good anyway. And these often have very strong smells because being pollinated at night, it's much easier for an animal to pick up a smell and follow it back to a flower than it is to see a visual signal at night and follow it back to a flower. So scent is a very important cue at night for animals that are trying to find, in this case, flowers. For bird-pollinated plants, Oftentimes, these first off have to have a lot of nectar because birds are very, very energetic. They burn a ton of calories. And for this activity to be worth their time, the plants have to produce a lot of nectar to feed those moths, or sorry to feed the birds like this hummingbird in this picture. They're often red colored because birds have a really nice ability to see into the red part of the visual spectrum. And these flowers actually do not typically have much of a scent at all because most birds, with the exception of a few things like turkey vultures, which are not pollinating plants, birds do not have a sense of smell, which is sometimes a little surprising to folks. They actually cannot smell anything at all, ever, with a couple of exceptions like some of the vultures. So again, it wouldn't make any sense for plants to invest heavily into very expensive, smell good kinds of attractants because birds wouldn't be able to smell them anyway. And all they would do is bring in insects to the flowers, which very likely wouldn't be able to pollinate to a very high degree anyway. So these plants are actually better off to do this attraction of birds with color as opposed to smell. For wind-pollinated plants, and there are still quite a few of these, flowers often tend to be very, very tiny. They're not going to have any nectar. They're not going to have much in the way of pigments. They're not going to have a smell to them because they're not trying to attract anybody. Their job is to produce as much pollen as possible and as many, effectively what amount to eggs as possible, to maximize the chance of wind-pollinated pollen getting to the right location, because chances are pretty slim it's actually going to make it. So they put all their energetic investment into simply producing a lot of pollen and then sort of hoping that some of it gets to where it needs to go. A lot of times these flowers are dioecious. These plants are dioecious, meaning there are separate male and female individuals. They're not monoecious or hermaphroditic. They're not both male and female at the same time. They often tend to be separate sexes. And a lot of times these plants also have flowers of a different type. You think of a flower like that, or like this one here, or these flowers. I mean, that's a really nice, kind of pretty, attractive flower. The flowers in this picture over here are catkins. And so they're a very different kind of a flower. They're very, very good at getting pollen distributed and getting pollen to be received as well by a female plant. But they're not very good in terms of attracting pollinators with smell or sight, for example. All right, so the benefits of pollination are several-fold, actually. The primary one being genetic diversity, because when you have different parents producing offspring through a process of meiosis, that gives us a lot of opportunities for genetic diversity to pop up. In environments that are relatively stable, outcrossing is not really all that necessary. In fact, if both parents are doing very, very well in their own environments, then both parents would be better off to reproduce by themselves because there's less variability in the offspring. Right? So if you have a stable environment that's not changing, and if you're well adapted to that environment, you might as well just more or less clone yourself, because then your offspring are going to be adapted as well. So that's kind of the second part of the question there too, is the environment stable? Reproducing sexually does have some risks associated with it when it comes to the genetics of the offspring, so it's possible that you'd be better off to be a bit more conservative and just clone yourself rather than trying to produce offspring through with the help of a mate. All right, so we're going to cut off here, and I will start up the next section here shortly.